in the marine invertebrates topic, come to the module 3, that is intertidal marine invertebrates. When we say intertidal, that means, as I explained earlier, it is the between the area between the highest high tide and the lowest low tide. And that means when the tides go down, these are exposed, and when the tides come in, these gets covered with the water, the sea water. And that means this is called as the convergence between of land, air and sea, and the organisms living this environment have to have the ability or the adaptive mechanism to sustain such kind of conditions. This environment is between high tide and low tide, the most physically severe environment on the earth, as we can say, because of these complexities involved. And the changes related to both climate and the human have their greatest impact in this particular habitat. If you see this particular image, what you can see is there is a spray zone or the splash zone, and then you have the high tide zone, the mid tide zone and the low tide zone. And if you go from this particular region, we have at the height, very highest high tide, or in the splash zone and the high tide zone, we will find mostly the shelled organisms. As you come down the depth or down the low tide zone, you will find mostly the soft bodied organisms or mostly the algae. And this particular habitat or the intertidal habitat has been mostly it is covered by the benthic, the sessile and the sedentary organisms. So benthic is nothing but, as I told you in the previous module, it is the organisms that live in the sediment or in the crevices or bores. Then we have sessile in the sense they cannot move. They are attached to a substratum holding fast with different mechanisms. For example, barnacle, they have a shell, basal shell plate, with that they attached to the adhere to the substratum. Then we have sea anemones, then we have different kinds of algae which are have the hold fast to attach. And then there are sedentary in the sense they have a very slow movement in this particular zone. So we have high diversity, organisms have low movement, then high adaptive mechanisms in terms of behavioral, then the morphological, then the physiological adaptations they have to take. So, and these can be these have recently, because studies have said that this can be used as indicators of the changes that are happening in the environment. So when it comes to most of the intertidal invertebrates, they have a biphasic life cycle. Biphasic life cycle in the sense when they are in the benthic phase, they have attached themselves to a substratum. That means they have a settlement process, then there is adhesion, there is a temporary attachment, then the permanent attachment, and then they develop themselves into an adult. That means all this phase takes place when it comes to the in the attached phase, that is the benthic phase. And whatever the developed adults, what happens is they release the larvae, and when they release the larvae, they are in the planktonic form. That means they release young ones, their larvae, in the water column, and those water column properties govern their successful development when it comes to the, and this phase is called as the pelagic phase. And this is this is almost thumb rule in most of the intertidal invertebrates that are inhabiting in this particular environment. So here I will give you an example of how the population connectivity of marine organisms is and why it is important. Because what you say here, you have a few adults, and those few adults will release a number of larvae. You can see a number of larvae, and adults have the capacity to release sometimes 5,000, sometimes 10,000, and sometimes more number of young ones into the water column. So there is adult and there is larvae, and in between there are a lot of processes that take place for the larval pool to get back into the adult pool. So what are the major constraints that are there? You have the major pool, the biggest part of this particular funnel, you have the larval pool. That means you have number of larvae, a high number of larvae. Then there are physical and larval transport processes. Physical is what? That means they can be carried away into the offshore region. They can carried out in the offshore region. They have to be in the onshore regions, and then there is a acting of with the time and space. These physical forces they determine how the larvae will be coming back to their local population or to the place where they have to settle themselves in the intertidal habitat. Then there are transport processes we have the currents. Sometimes they may be along shore, sometimes they may be perpendicular to the shore, and this determine their fate bringing them back to the settlement process. And when they are near the settlement habitat, or once they are there in the settlement habitat, the microhydrodynamics that are acting at the substratum, and also the behavior and the substratum availability. It is not that there is a process that any substratum they will come and settle down. They require a conducive substratum. They require some chemical signatures to attract them to the substratum. And mostly, if I give an example of barnacles, there is a process called gregariousness. There is nothing but the larvae will try to find its own conspecific. That means their own adults or own young adults to settle near them, because there is a cross fertilization is thumb rule in these particular organisms. If I give an example of barnacle, that means they require an adult of their own conspecific. And once they are near their substrate availability, then the local biotic interaction and the disturbances will determine how the adult population grows. And taking cue from this, we carried out some observations how the larvae are dispersed in the Goa coast, along the Goa coast. And we took a domain from Arambol, that is in the north portion, to Galji Bay, which is almost south of the coast of Goa. And what we did is we took some these are the three places as the spawning grounds. Spawning grounds are nothing but there are adults situated there, and they release the larvae in the planktonic environment or in the pelagic environment. And we took during 3 different seasons, that is northeast, then pre-monsoon, and during the post-monsoon season, because the wind or the local winds or the breeze that is acting during these seasons are entirely different. For example, during the northeast monsoon, the transport is along shore. During pre-monsoon, the movement is southward, and during post-monsoon, there is difference. There is southwest winds in the first half, retrieval of the monsoon, and then it is transport. During the onset of the northeast monsoon, it is major north and minor south. So what we found is, and there is a movement. If you see this particular movement or a movement from here to here, you will find the larvae can move along the shore up to nearly 70 kilometers. That means they are dispersed from their parent animal or from the spawning ground to 70 kilometers. So during this process of transport, either they have to find a competent substratum. The competent larvae have to find a suitable substratum to settle themselves and then grow into an adult population. So this is a mechanism which governs the population back in the marine environment. And after this, it is very important how these organisms can be used for ecosystem analysis. And so the title I have given is Ecosystem Analysis, Utility of Invertebrates. How you can utilize these to study what happens in the ecosystem functioning. So I took example here is a barnacle, which is a very important marine invertebrate and also a fouling organism when it comes to invertebrates. And they also have a biphasic life cycle. What you see here is the naupliar phase, which have to be which are dependent upon the phytoplankton for their food, and this is the benthic phase, wherein you have cyprid. This particular larvae is specialized for getting going and settling and recruiting itself into a juvenile and then grow into an adult. So during the observation, what we did is we studied how much larvae are coming and getting recruited in the natural population. So during this process, during the entire study period, we just went and scraped some part of the concrete structure, and then try to count the how many have been getting recruited. And once the study is over, we saw that there is a varying pattern in the recruitment of these barnacles. That means whatever larvae are there in the environment, all will not come and get settled on on that particular substratum. And we related it to also because we are in a tropic condition, and while there is a range for about 4 to 5 months, we find there are breaks in the monsoons. And during these breaks, it is seen that there are phytoplankton blooms that occur. Phytoplankton blooms will support the pelagic larvae because they feed on the phytoplankton, and when they grow fast and again settle, and but after the break of monsoon, there are recurrence of again spells of the monsoon. There are rainy spells, and this puts the whatever the settled population into a risk, and there is a heavy mortality that has been observed soon after this break. That means what? There is a imbalance. There is a imbalance in, or there is a what you can say, there is a mismatch of this particular cues. That is mismatch of the release of larvae because of there is lot of food, and there is recurrence of monsoon again. That means there is population comes in, they feed, they grow, they settle, but the settled population doesn't recruit and grow into an adult. So this is a kind of impulsive luxury, what you can say. So it is a short-sighted luxury. So what I coined this is life in tropics, not at that all that easy and synchronized. While life in tropics, because in the temperate condition we don't find such kind of phenomenon of occurrence and recurrence of monsoon as we find in the tropics. And while coming to ecology of sessile invertebrates in the environment, what I did some experiment is, you can find there are organisms sitting in this particular zone. You have the splash zone, the surf zone, the top one, then we have the Balanus amphitite in the middle, and in the Megabalanus intinabulum, which is almost in the down portion of that particular intertidal zone. And I've presented a result of their mortality at 35 degrees centigrade under desiccation conditions. Means they are exposed to the sun, they are not in the water column. And what is interesting to see is, you see this green line, this belongs to the organism that is sitting at the top. That means the one that is at the top of the intertidal region survives for the longest duration. It means what? That it has got some physiological adaptation to sustain themselves in this particular zone. So can be used as potential biomarkers for changing the environment, we can say in simple terms, the climate change. So if you see this, I will just quote this, the climate model, which is having the sea surface oxygen, then the global warming predictions, and also the pH. So if you see this particular and you see the trend that is there, and whatever the predictions they have made for next 100 years, they have said that there will be global warming, there is warming of sea surface temperature, then increase in the pH. Taking cue from those particular predictions, we did some laboratory studies with using a marine invertebrate. This is showing present condition and future conditions, that is at 30 degrees centigrade, 400 microatmosphere of CO2, and also at future condition, that is 34 degree and 1500 microatmosphere of CO2. And what it shows is, with at this particular conditions, you will have, if you see this particular region where you have already settled some juveniles of organisms, and you have the larger forms of diatoms in the biofilm. But with the future conditions, with increase in the pH and also the temperature, what you can say, you will find there are so small tiny organisms that are there that occupies that particular habitat, that is in the intertidal habitat. That means the one which are providing cues to these intertidal organisms in that particular zone, there will be a mismatch, there will be a difference, or there will be alteration in the mechanism that they come and settle. So climate change impact the biology of intertidal organisms, viability of algal spores, and also will alter the settlement and recruitment pattern. And how climate change would influence these intertidal invertebrates. So that is the future work to be carried out, and we have a lot of avenues to study is in the future. With this, I come to the end of module 3.